Great. Our next speaker is Jamie Lee from Imperial College London.
Wait, sorry. Hi, thank you for this opportunity to present my work today. My name is Jamie and I'm a PhD student at Imperial. And today I'm going to be presenting my PhD work where I'm looking at the role of the skin microbiome in the pathogenesis of AD. And I'll be talking about modeling the microbiome battle on AD skin and the effect of promising therapies. So AD is a super common inflammatory skin disease that affects 1 in 5 children worldwide. Its symptoms include super intense itching and painful lesions. And despite the therapies currently available, there's still a lot of people who struggle to manage their symptoms. So the pathogenesis of AD can be distilled into these 3 key factors. So there's skin barrier damage, immune dysregulation, and microbial dysbiosis. And these 3 factors are really strongly interlinked. So for example, if there's skin barrier damage, then that can encourage microbial dysbiosis or the proliferation of a specific pathogen, such as Staph aureus in the context of AD. And Staph aureus can produce these toxins that damage the skin barrier and increase inflammation. And the dysregulated immune response in AD patients can further increase barrier damage. So this makes it a really difficult cycle to get out of. So overall, there's still a lot that isn't really known about how the skin microbiome and the host interact with one another. But for my current project, I'm focusing on the interactions between the microbes on the skin and how it affects the skin barrier integrity and how this can help maintain skin health or exacerbate disease. So first, what does the microbiome composition look like in healthy and AD patients? So the microbiome composition is super different in healthy and AD patients, where healthy patients tend to have a very diverse microbial composition and tends to lack Staph aureus. And in contrast, AD patients tend to have a very collapsed microbial composition, and S. aureus tends to be the dominant species. But we can see from here that S. epidermidis also seems to be a pretty key player. So if we go back to our plot of the healthy patients and just focus on the Staph species on there, we can see that S. epidermidis is also really dominant in some of them, which raised the question of whether we can capitalize on the presence of S. epidermidis in AD skin to help us move to a healthy state and how we might be able to do this. So the main research question that we're focusing on is how we can use epidermidis to help us move to a healthy state. And to look at this question, we came up with smaller sub-objectives. So the first one was to develop a model of the dynamic interplay between S. aureus, epidermidis, and skin barrier integrity. And then after that, we wanted to analyze the system to understand what steady states the system converges to. And then after gaining a better understanding of the system, we apply a hypothetical S. aureus specific killing therapy, which is believed to be like a promising treatment strategy to see what actually happens when we do apply it. So first, in terms of the model formulation, this is our system of 3 ordinary differential equations. And we're looking at the interaction between 2 key bacteria on AD skin and its interactions with the skin barrier. So there are a couple of key points that went into the development of this model. The first one is that both S. aureus and epidermidis have the capacity to produce these toxins that directly cause skin barrier damage. But the damaging properties of S. epidermidis is kind of a new area, so not everybody is affected by it. And then we also said that the skin barrier integrity limits like the nutrient availability to microbes on the skin. So like if the skin barrier integrity is high, we said that these microbes won't proliferate as quickly, whereas when the skin barrier integrity is low, there'll be more nutrients available to these microbes on the skin that will allow them to grow and proliferate. And then the final point is that these two microbes can also produce these antimicrobial peptides that kill one another off. Another thing that we included in the model was the Quorum sensing system that S. aureus and S. epidermidis use to produce these toxins or antimicrobial peptides. And it's basically a population-dependent system where below a certain density threshold, these bacteria aren't going to invest in the resources to produce antimicrobial peptides or virulence factors. Whereas once you cross this threshold, then they do. And we modeled that really simply as, but described in the image. And then the final point is that we tried to estimate as many of the parameters as we could directly from literature, and we managed to estimate 9 out of 14 of those, where one parameter set would represent one virtual patient. But obviously, because of the huge uncertainties in the estimated parameter values, we basically explored lots of different parameter sets by randomly sampling over like a specified distribution of the parameters. And we found that all of the patient types that we saw could be grouped together. So for the rest of this presentation, I focus on one particular parameter set, which represents one virtual patient. So from the steady state analysis that we did, we saw these 3 stable steady states. So we have 2 stable steady states where we see a coexistence of S. aureus and S. epidermidis. And we have 1 stable steady state where we see kind of the dominance of S. epidermidis. This plot is a bit confusing because when it's at 0, I actually mean like a 0 population size. So it's a complete dominance. And we wanted to see how closely this reflects clinical data. So the clinical data that we have is from 13 patients with AD, and they took swabs from lesional and non-lesional sites, so that corresponds to a disease or a healthy state. And yeah, they estimated the population sizes that they saw from the swabs. So we can see that the clinical data is pretty different from the model, but there are some key qualitative characteristics that I feel we can capture. So the first thing is that at a healthy state, you tend to see lower population sizes of S. aureus and S. epidermidis, whereas when you're at a disease state, there tends to be a higher population. And then we can also see that like with the stable steady state where S. epidermidis is dominant, there are a couple of patients with that characteristic here. But the most important thing that we do feel like the model can capture is the coexistence of S. aureus and S. epidermidis in AD skin. So then we wanted to understand how this coexistence comes about in a diseased and healthy state. So with the disease state, there's strong competition between these 2 bacteria, and we wanted to look at the mechanisms for how they coexist. So to do this, I created lots of different virtual patients. So this plot shows 1,000 example virtual patients where the parameter values are picked from specific distributions. And we found that with all of these virtual patients that we saw, there was always going to be a stable state in the healthy region and in this region here. But for these 2 stable states, you would either have the coexistence of S. aureus and S. epidermidis, or you would have an overgrowth of S. aureus. So we found this like trail-like behavior quite interesting because the S. aureus population size stays pretty constant, but there's a pretty big variation depending on what parameter sets we were looking at. So then we wanted to look at the sensitivity of the S. epidermidis stable steady state based on changes in parameter values. So that's this here. And we essentially found that there were three main parameters that were important in maintaining this coexistence between S. aureus and S. epidermidis. And that's the strength of killing of S. epidermidis by S. aureus. So that has to be relatively weak compared to our nominal parameter value. And the growth rate of S. epidermidis has to be relatively high, whereas the strength of the inhibition the strength of the inhibition by the skin barrier needs to be relatively low. So it comes from a balance of these three things. And then now moving on to the coexistence at a healthy state. This is somewhat more intuitive because these two bacteria aren't really competing with one another at a healthy state. So the steady states of these two bacteria are reached independently. And the coexistence comes about from the balance between the skin barrier inhibition by the skin barrier, the growth rates of the two bacterial populations, and the killing of the bacteria by skin turnover. So after getting a better understanding of how the mechanisms in our model for how coexistence comes about, we wanted to see how we should develop therapies to move patients to a better state. And we wanted to think about whether the healthy state is actually a state that we would want to shift patients to. And we felt, so to do this, we looked at the basin of attraction for these stable states. So in these plots, I started the ODE simulations from different population sizes and looked at where the steady states they converge to. So all of the initial conditions in this region would converge to here and so on. And we found that it's actually really easy to move out of the healthy state and into a disease state. So which suggests that if there's a small perturbation in the system, then it's really easy to fall into the disease state and really difficult to get out of it. And another thing that I found from the analysis is that the convergence to the healthy state is really slow, which means that if we have accumulating perturbations, then it's more likely to move out of it again. So we decided this isn't actually a healthy state that we want to move to, and the presence of any S. aureus on the skin will eventually cause us to shift back to a disease state. But this is still very much ongoing work. So the therapy side of it is kind of pretty new. So then the final bit that we looked at was we wanted to look at applying an S. aureus specific killing therapy to this example patient, because that's been hypothesized to be a promising therapy treatment strategy for many years. And we found that in this particular case, S. epidermidis takes over and continues causing barrier damage. So the patient remains in a disease state. But if the patient is colonized by a strain that isn't pathogenic or doesn't produce these virulence factors that cause skin barrier damage, then it's actually pretty representative of what we would see in healthy patients, where you tend to see an abundance of S. epidermidis, but no S. aureus. So something that we're interested in looking at is trying to develop therapies that target S. aureus, but also address the potential, address the fact that there could be other pathogens that could overgrow and damage the skin barrier, such as in the case of S. epidermidis. So that's kind of something that we'll look at next. So just to summarize, so far we've modeled the dynamics of a collapsed microbiome of AD patients, and we found that the model manages to capture this coexistence that we see in clinical data, and that the coexistence arises from different mechanisms. And we're currently working on developing new therapies that target S. aureus to treat AD. And with that, I'd like to thank my supervisor, Reiko, and 2 postdocs in the lab, Ahmed and Tapir, who are really important in the development of this work. Thank you for listening.
Questions?
You were saying that in the healthy state, these 2 strains aren't competing. Is that kind of an accurate representation of what we expect on this strain? Because I imagine that there's some sort of competition for space or some other reason.
Yeah, so it seems like on skin, it's actually not that easy for bacteria to grow. And from our conversations with like clinical collaborators, it seems like S. aureus needs different nutrients compared to S. epidermidis to grow. So we assume that there isn't a massive competition between the 2 of them.
Yes.
Some of the other work that I know about the microbiome has emphasized the importance of like the physical structure of the pores, how that makes it really hard. Once something kind of colonizes that, it's hard for other things to invade that. I guess my first question would be then, how does that pore structure change between AD and healthy skin? And what role do you think it does or does not play in the disease here?
Yeah, that's an interesting question. So I actually haven't looked too much at the pore structure, but that would be something to consider, because I know that like they say that like biofilm formation is a really common thing within pores, and that can kind of change the can kind of change like what results we might expect to see. So I don't have a good answer to that. But
Orkan.
Related to that, is the model spatial? And if you were to make the model spatial, how would you expect it to change?
Yeah, so right now the model isn't spatial, so we're assuming that everything is well mixed. It would be interesting to look at the spatial component of it. And I don't know, I wonder if we look at like a small enough region, we could assume that it's well mixed. But I guess like once we expand it, then yeah, we'd have to think about how things change.
Stephanie.
You might end up in different regimes, and also the parameters, the basins of attraction could look different, and perhaps then switching from the healthy to the unhealthy phenotype is not as easy or not as difficult as you would see in the well-mixed situation.
Right.
It's also more a comment than so.
Yeah. No, that's a good thing to consider. Yeah.
Okay. Let's thank Jamie and all speakers of this morning's session. We have we don't have the 3rd speaker. We don't know where he is. So we have early lunch. I mean, maybe Orkan has an announcement.
Yeah, actually, so we do know where he is. He's on a plane to here. But again, there was a hiccup and he thought he was speaking on Friday or something. So we'll juggle things around and slot him somewhere. But for today, we get an extra time before lunch. So make good use of it and discuss. Seb has something to say.
So we now have allocated VM to everybody, and we would like everybody to try and connect with VM, because we are not going to do it during the tutorial, because the tutorial is not on how to connect to that. Yeah. So that's it. So can you stay for like 10 to 15 minutes?